Okay, so last week we ended the class and I asked the question, would this molecule be soluble or dissolve in water? And the answer is no, because we do have a lot of this hydrophobic hydrocarbon here, and we do have this carboxylic acid, but this molecule is largely hydrophobic, so it would not dissolve in water. And this molecule is what we're going to talk about today, which is that it is a fatty acid. So we're going to begin talking for the next few lectures about biomolecules, and then we'll end with um, metabolism. And to give an introduction to biomolecules, biomolecules are um, molecules that are present in organisms that are essential to one or more typical biological processes. And in this class, we'll be talking about lipids today, which consist of oils, fats, and waxes. We will be discussing in the next lecture carbohydrates. These are starches and sugars. We'll also then be talking about proteins, and then we're going to end with nucleic acids such as DNA and RNA. One thing that we will also be talking about and we'll see a lot are some monomers and polymers in biomolecules. So monomers are one, and so we will be talking about, for example, DNA, and those are large, carbon compounds, but those large carbon compounds are made from smaller, simpler molecules, and those simpler molecules are called monomers. And then those monomers can bind to one another to form complex molecules known as polymers, poly meaning more than one. We're going to talk about lipids today. We're going to start with the monomer unit of some lipids, and then we're going to see how those different monomers can come together to form some more complex structures or polymers. So specifically for lipids, lipids are hydrophobic nonpolar molecules, which we saw in our first slide. They are soluble in nonpolar solvents, and they are insoluble in polar solvents such as water. The types of lipids that we'll be looking at today are fatty acids in the carboxylic acid family. We'll be talking about triglycerides. This is a polymer, so it'll be made up of 3 fatty acids with a glycerol backbone. We'll then be talking about phospholipids and glycolipids, and this is again a polymer. It's made up of some fatty acids, a glycerol backbone, and a phosphate. Steroids, which are derivatives of cholesterols. And then we'll talk about our biological membrane and how these lipids function in our cells. We're first going to start with fatty acids, as these are the monomer groups. Fatty acids contain a carboxylic acid group, and this should make them polar. So that's that one tail group that we saw on the left side of the molecule. However, they also contain this very long hydrocarbon tail. And because of that hydrocarbon tail, that's what makes the molecule overall nonpolar. So a question that you could get asked on exam is, would this molecule dissolve in water? And you might want to say yes, because it does have that polar group on it, but because the molecule is largely a hydrocarbon, it will be nonpolar. This is what a typical fatty acid looks like. Fatty acids have between 12 and 20 carbons, and the number is usually always even. And when we talk about metabolism in our last lecture, you'll kind of see why, and it's because of how it's synthesized and also how it's broken down, that they tend to be even numbers. So we're going to describe a fatty acid in these different regions. We're going to say that this long hydrocarbon here is the nonpolar tail. And then we have this carboxylic acid group, which we should recognize from our lecture last week, where we talked about organic molecules and those functional groups. but this is a C with a double bond O, and then it has this alcohol group making it a carboxylic acid, and this is going to be its polar head group. And these non-polar tails interact with each other through London dispersion forces. And those London dispersion forces are very weak, but they are interactions that do occur and impact how fatty acids behave. Fatty acids can contain these long hydrocarbons that either have single bonds, so those alkane groups that we learned about last week, or they can have double bonds, and those are those alkene groups that we talked about. And when we have a fatty acid that doesn't have any double bonds in it, we're going to say that it is saturated. And when we have a fatty acid that does have double bonds, we're going to say that it's unsaturated. I'm going to just answer a question here. Is the chain the same as the one you were speaking about in the beginning of a lecture from the picture when you were picking up from last week? Yes. So that long group is that fatty acid chain. So you can have just one unsaturated fatty acid. So we would call that monounsaturated. So here is oleic acid, and this has a double bond here. You can have also be polyunsaturated. So linoleic acid has two double bonds, and then linolenic acid has three double bonds. And you can see that there is a change in the chain structure if you add some double bonds in. So you go from this very linear and straight molecule to something that has a bit of a bend in it. And this bend is going to impact how these molecules can stack on top of each other. And that's going to impact those London dispersion forces. So here are some common fatty acids that you see. You may have all be familiar or have heard of omega-3 fatty acids. So linolenic acid is one of the omega-3 fatty acids. So in this table, you can see the number of carbon atoms. These all have an even number. You can see the number of double bonds. So if it has a 0, then it's saturated fatty acids. If it has a 1, it's unsaturated. And then we'll talk about these melting points. And here are some sources of where some of these fatty acids come from. So if you use olive oil in cooking, then you have the fatty acid oleic acid is found in olives. So as I mentioned in the previous slide, the cis double bonds produce these bends here in unsaturated fatty acids, and this prevents these molecules from stacking on top of each other, and it disrupts the London dispersion forces that these hydrocarbon or nonpolar molecules can have. So on the left, we're looking at stearic acid. It is a saturated fatty acid. And so it doesn't have any double bonds. It doesn't have that bend in it. And so these molecules can stack on top of each other. And those weak London dispersion forces are what are going to hold these molecules together. And because of that, saturated fatty acids tend to be solids. And one way that I've always remembered this is S is for saturated. Solid is for saturated. On the other hand, we have linolenic acid, and this has these double bonds that give it this bend. And because of that bend, these fatty acids don't stack as nicely on top of each other. And because of that, those London dispersion forces are disrupted. So unsaturated fatty acids tend to be liquids. So we can see this here, that here we're showing a graph where we have the melting temperature on the y-axis and the number of carbons. So as we increase the number of carbons, you have to heat up the molecule more in order for it to melt. So you would say that this is more of a solid, and that's because you have that stacking. So when you get to about 18 carbons, you have more of those carbons where you can stack on top of each other. When we look at double bonds, though, we can see why they tend to be liquids at room temperature and not solids. And it's because of those double bonds. So here, instead of the number of carbons on the x-axis, we're actually looking at the number of double bonds versus the melting temperature. So as we increase the number of double bonds in the fatty acid, you can see the melting temperature decrease. And so at this point, you pretty much have a liquid. So next we're going to talk about triglycerides. So we started with our monomer, which is our fatty acids, and now we're going to talk about some polymers here. So triglycerides are a storage form of fatty acids in mammals. So if you've ever had blood drawn and they look at your, fatty acid levels, they're typically looking at your triglyceride levels. And high triglyceride levels in the blood are at risk indicator for arteriosclerosis. And here is just some data from the American Heart Association, just talking about what some normal levels, borderline and high levels of triglycerides are. Triglycerides are a combination of three fatty acid molecules with a glycerol molecule. So this is a very basic structure. The tri means three. And so we have three of those fatty acid molecules that we just saw, and they are bound to this glycerol, what we call a backbone. You can kind of think of it like an E structure. And this is more of a molecular level where you can see these very long hydrocarbons, so these fatty acid residues. And here, where that carboxylic acid is, you actually have another functional group, which is called an ester. So you have that oxygen that's sandwiched between these two carbon molecules. And that is what attaches these fatty acids to this glycerol. And this is how your body likes to store fat. And depending on which molecules that are attached, fatty acids, they don't all have to be the same. So all triglycerides don't have stearic acid attached to them. You can have different fatty acids that are attached to this glycerol backbone as well. So just as with fatty acids, the presence of cis double bonds lower the melting point. So triglycerides made from unsaturated fatty acids have lower melting points than those made from saturated fatty acids. Triglycerides from animals tend to have a higher proportion of saturated fatty acids. I'm going to pause here to take a question. So which part would you like me to explain a little bit more? Should I go back to this one? Great, yes, I can do that. So what we're looking at here is a triglyceride, and this blue part is just the fatty acids that we discussed in the last slide. A triglyceride is going to have three of these fatty acid residues here, and they're going to be bound to this molecule. So this CH2CH2 is called glycerol. And so the name comes from three fatty acids that are attached to this glycerol residue. And the way that the fatty acids attach is through that carboxylic acid group that was on the end, that polar head group that was on the end of the fatty acid bonds is what is attached to the glycerol residue. So what you want to look for, let's say on a quiz, you might get asked to identify what fatty acid this is. Um, you want to look for essentially like this E structure where you have this backbone and then these three fatty acids here. And this might look different, particularly in the fatty acid residue. So these don't always look the same. These might be different. You can have two of the same fatty acid residues and one that's different. I'm going to show in a few couple of slides that you can have unsaturated fatty acids here. You can have saturated fatty acids here. So this part is variable, but a triglyceride will always have three fatty acids attached to this molecule here. Is that clear, or should I explain a little bit more? Yeah, we can. I'll go through some more, and if you have any questions at the end, um, we can address it. But you can think of it as in these fatty acids are monomers, and they come together to build this polymer structure. So again, you can have unsaturated or fatty acids that have double bonds attached to that glycerol backbone, and triglycerides from animals tend to have a higher portion of saturated fatty acids. So because they're saturated fatty acids and they're linear and straight, they can stack on top of each other, and solid equals saturated, or saturated fatty acids are solid at room temperature. So we call these fats. And for example, butter, lard, and baking grease are triglycerides that originate from animals. Um, and then triglycerides from plants tend to have a higher proportion of unsaturated fatty acids. So remember, those unsaturated fatty acids have those double bonds in them. And because of those double bonds, they can't stack on top of each other. So most are liquids at room temperature, and we call those oils. So takeaway from this slide is saturated fatty acids are solids, and those are fats. And then unsaturated fatty acids are liquids, and those are oils. Fats tend to originate from animals, and oils tend to originate from plants. So now that we've covered triglycerides, we're going to kind of build on that concept, and we're going to talk about phospholipids and glycolipids. And these are very, very important because this is what biological membranes are made of. So our cells have a membrane around them, and you can find phospholipids and glycolipids here. We're going to talk about something called being amphipathic. If you use hand soap or bar soaps, these molecules are amphipathic. And essentially, what that means is that these molecules have a polar component as well as a non-polar component. And this is what makes our lipid bilayer. We are going to take a closer look at that in this slide here. So this is what soap molecules look like. We call these micelles. And essentially what happens is that we have these blue molecules here. These are polar. And these brown kind of tan strings are nonpolar. And when you put these molecules in water, these molecules are naturally going to form a micelle where they hide all of the nonpolar components in the middle and all the polar components outside. So what you want to think about is that polar parts can interact with water, non-polar parts can't. So you want to hide those non-polar parts on the inside and only have the parts that can interact with water on the outside. This is why soap works in removing dirt, because the dirt or non-polar things are attracted to this non-polar component of the micelle, and then the water, the polar components are attracted to the water. So when you wash your hands with soap, you get all the dirt and grime off of them on the non-polar part, and then you wash it away with water. So that polar component is attracted to the water. So you get it all off of your hand. That's how soaps function. Your body does something very similar in its cell membrane. The purpose of the cell membrane is to keep stuff out of the cell that we don't want to be out. So it forms this barrier. So phospholipids, as we're going to see, are going to have this polar head group, and it's going to have these hydrophobic tails. And these tails, similar to a soap molecule, are going to be inside of the bilayer. They're going to be protected because we don't want the nonpolar parts to interact with the water, but these polar head groups are going to be facing the water. So always remember, we want the polar parts of a molecule to be facing water molecules because they can interact, and we want to make sure that our hydrophobic parts are hidden on the inside. Are there any questions on micelles and kind of how we want a molecule that has both polar and nonpolar parts? We want to hide those nonpolar parts in the middle. And I'm happy to take any questions afterward, after you spend some more time looking at this. But non-polar parts on the inside, polar parts on the outside facing water. So there are two types of phospholipids. We're going to talk about glycerophospholipids. So the reason why we talked about triglycerides first is because now we're going to talk about molecules that look similar to triglycerides, but just a little bit different. So we're going to still have our glycerol backbone. We're going to have two fatty acids, but instead of that third fatty acid, we're going to have a phosphate group and an alcohol group. And we'll take more of a molecular look at that in the next coming slides. So phospholipids, glycerophospholipids have glycerol, two fatty acids, and a phosphate. That's where the glycerol phospholipid comes from. Lipid because of the fats, glycerol because of this glycerol, and then phospho because of the phosphate. Next, we're going to have a sphingolipid. And so instead of the glycerol, we're going to replace the sphingosine, which you don't need to know the structure of, just know that sphingosine replaces the glycerol and you have this fatty acid here. And then you also still have a phosphate group. So this is why it's called a phospholipid, because you have this phosphate here still. And this is the same glycerol phospholipid, just talking about how there's usually an alcohol attached to the other side of this phosphate here. So this is what this molecule, this sphingolipid looks like. It functions similarly to a glycerol phospholipid, but it's structurally different. Again, there is no glycerol here. So where you would have those three carbons in that glycerol lipid or triglycerol, you're going to have this sphingosine molecule. You don't need to have any of this memorized. You don't need to be able to draw this. Just know that you don't have glycerol, you have sphingosine in this molecule here. And the importance of this is that sphingolipids are found in the myelin membrane that insulate the nerve cells. Some sphingolipids use sugars for the alcohol portion of the molecule. So in this, oh, sorry, glycerol phospholipid, where we have this alcohol here, and a sphingolipid, it could be a sugar molecule. But again, you don't need to um, memorize that. Um, if you have a sphingolipid that has a sugar, these are called glycolipids. And this will be more clear once we cover our carbohydrate lecture. But the importance of sphingolipids are that they are very important in the brain and protecting our nerve cells. Next, we're going to talk about steroids, and steroids are a type of lipid that are not derived from fatty acids. So we're not going to see those long fatty acid chains attached to any molecules. Instead, we're going to look for some ring structures. So we learned about some cyclic molecules in our organic molecules lecture in our functional group, and steroids are going to look very similar to that. So all steroids are based on a 5-cyclohexalkane ring, and these rings are all fused together. So we're going to take a look at some of these molecules in a few slides, and you'll notice that they all have this ring structure. Instead of these molecules being derived from that like very long fatty acid, our steroids are made from cholesterol. So, you want to limit the amount of cholesterol in your diet, but it is also very important to have an adequate amount of cholesterol, because without cholesterol, you cannot synthesize your steroids. So this is what cholesterol looks like. And if you note, it has that fused ring system that we see in our steroid molecules. So this is going to be the precursor to that. Cholesterol is only found in animals. And besides being used to synthesize the other steroids, cholesterol is dissolved in a membrane to keep them fluid. So we're going to take a look at the plasma membrane of the cell, and you'll see that there's some cholesterol molecules, and I'll explain why that is. Since cholesterol is only found in animals, you might ask, what do plants use? And plants use the alternative strategy of using polyunsaturated fatty acids to make their phospholipids. So they don't use cholesterol. So these are some common steroids that either can be found as biological components of our cell membrane, or you've heard some of these, such as testosterone, cortisol, corticosterone, corticosterone, estradiol, which is a precursor to estrogen, progesterone. You may have heard some of these, but again, you can see that they all have this ring structure here that is fused, and they all look similar to cholesterol because that is where they originate from. So let's take a look at the functionality of what we just talked about. In our metabolism lecture, we're going to talk about the use of fatty acids as energy, because your body can burn them to produce high amounts of energy. But in this lecture, we're going to talk about our cell membrane and this fluid mosaic model that we see. So our cells have this lipid bilayer, and we see that it's made of multiple components. The purpose of this bilayer is that we want to be selective to what molecules we want to let in and what molecules we want to let out. Everything cannot pass through our cell membranes, and that's a good thing. We don't want just anything coming inside. So here's a zoom up on a phospholipid. We have that phosphate group, which is going to be polar. And then we have these fatty acid tails. Those are those long hydrocarbon chains that we looked at the beginning of the lecture. And just like we talked about with soap, it's going to form this bilayer where you have your phosphates facing the outside, so where water is, and then your cells also have water on the inside. So the phosphate groups are going to be facing where the polar parts are of the cell. And then we're going to hide those lipid molecules, these non-polar fatty acid tails on the inside. Now, this would kind of prevent some molecules to come through. And we're going to talk about in our protein lecture why it's important to have proteins embedded in the bilayer, because we need to allow things inside of the cell. And these proteins are going to allow that to happen. Now, in addition to our phospholipids forming this nice bilayer, we're also going to have cholesterol. And cholesterol is going to be found in between these fatty acid tails so that they don't stick together. We don't want our cell membrane to be solid, right? Because if we have saturated fatty acids, we're going to be have solids, and we don't want that. So we want to make sure that we have some molecules that can kind of disrupt those London dispersion forces if they happen, and keep this membrane fluid so that we can allow things in and out. And then this is a glycoprotein, and we'll talk about this in our carbohydrate lecture. So this is the biological significance, or one of the biological significance of lipids, is that they're very important in our cell membrane. And what I like for you to remember from this slide is just that you have these phospholipids that form your bilayer, and you have cholesterol in between that allow the membrane to be fluid. So I'm going to ask some questions here, and you can put the answer in the chat. What group of lipid that we talked about does this molecule belong to? And we spent quite some time on this. Anyone want to give it a try? It's got this. Yep. Yes. So we have one triglyceride. Wonderful. And the way that you know that is because you have this backbone here, this glycerol backbone, and these three fatty acids that are attached. And I always remember because it kind of looks like an E here. Okay, the second question is, and I gave a little hint here, what group of lipid does this molecule belong to? Yep. Yep, so this is a glycerol phospholipid. So we see here that we have this glycerol, we have our two fatty acids, and then here we have this phosphate group here. So phosphate is this P, and then it'll have this double bond O, O, and O here. And instead of there being an alcohol group, we'll talk about what this molecule is in a few lectures. But the important part to look for is this glycerol, these fatty acids, and then this phosphate here. And then lastly, an easy one, saturated fatty acids are liquid at room temperature, true or false? Correct. So it's false, right? Exactly. So because they are saturated, um, saturated fatty acids are solid. So saturated, solid.